Cells break down glucose and derive energy from it by using an oxygen-requiring pathway called cellular respiration. Cellular respiration has four phases. The breakdown of glucose begins with glycolysis, which occurs in the cytoplasm. The next three phases of cellular respiration, the transition reaction, the citric acid cycle, and the electron transport chain, all occur within mitochondria. In glycolysis, glucose, with 6 carbons, is split into 2 molecules called pyruvate, with 3 carbons each. Glycolysis occurs in several steps, each of which requires a specific enzyme. During the first steps, 2 molecules of ATP are consumed, providing glucose with the energy for splitting. During the remaining steps, 4 molecules of ATP are produced, for a net gain of 2 ATP. Glycolysis also delivers energetic electrons from glucose to molecules called NAD+, forming the energy-carrying NADH molecules. Note that glycolysis does not require oxygen. The pyruvate molecules from glycolysis move from the cytoplasm into the inner compartment of the mitochondrion. Let's look at the fate of just one of these molecules. Pyruvate reacts with a substance called coenzyme A, CoA, in a reaction called the transition reaction. The transition reaction results in the removal of 1 carbon in the form of carbon dioxide or CO2 from pyruvate. The resulting 2-carbon molecule called an acetyl group then binds to CoA to form acetyl-CoA. A molecule of NADH is also produced from each pyruvate. In the next set of reactions called the citric acid cycle, also called the Krebs cycle, acetyl-CoA is stripped of its final carbon atoms in the form of carbon dioxide. During these reactions, energy is captured and stored in ATP, NADH, and another energy-carrying molecule called FADH2. The cycle begins when acetyl-CoA reacts with a 4-carbon compound to form a 6-carbon compound. By the end of the reactions in this cycle, the 4-carbon compound is regenerated and can participate in another round of reactions. The NADH and FADH2 produced in the previous phases enter the electron transport chain, the final phase of cellular respiration. A series of carrier proteins, the electron transport chain, embedded in the inner membrane of the mitochondrion, accept the high-energy electrons from NADH and FADH2. Eventually, the electrons are passed to oxygen, the final electron acceptor, which then combines with 2 hydrogen ions to form water. Without oxygen, the transport chain comes to a halt, blocking the citric acid cycle and stopping cellular respiration. During the transfer of electrons from one protein to the next, energy is released and used to make ATP. Altogether, cellular respiration produces 36 molecules of ATP per molecule of glucose, 2 ATP from glycolysis, 2 ATP from the citric acid cycle, and 32 ATP from the electron transport chain. Although cellular respiration requires oxygen, cells can still harvest a small amount of energy from glucose when oxygen is scarce. Fermentation is the breakdown of glucose without oxygen. It begins with glycolysis, which produces 2 molecules of pyruvate from a molecule of glucose. Importantly, it results in a net gain of 2 ATP and 2 NADH molecules. The remaining reactions of fermentation also take place in the cytoplasm, transferring electrons from NADH to pyruvate. In this example of lactic acid fermentation, which takes place in muscles during vigorous exercise, the result is 2 molecules of lactate. This reaction is critical because it regenerates NAD+, which is essential for the production of ATP through glycolysis. Fermentation is a very inefficient way for cells to harvest energy. It nets only 2 molecules of ATP, compared with the 36 molecules of ATP produced by cellular respiration.